Hello, my name is Jasmine Norris. In this video, I will be comparing a news article to its primary source. Both sources discuss the cancer resistance found in elephants and how it compares to humans. First, I will provide a summary of the news article. The news outlet that this article comes from is very well known, and it is called CNN. The title of this article is Elephants Rarely Get Cancer. Here's why it matters to humans. And it was authored by Michael Nadelman. The article starts off by stating that many would assume behemoths, such as elephants, would be very cancer-prone due to the vast amount of cells that compose their bodies and their long lifespans. This was disputed, as less than 5% of elephants die from cancer, as opposed to the 25% of humans who do. The reasoning for this lies behind their genes, dubbed zombie genes. These genes cause cell death in order to combat the damage in DNA. The gene responsible for this is known as p53. It is known as the tumor suppressor gene. The human genome possesses 2 copies of this gene, but elephants have dozens. These findings proved that body mass and cancer vulnerability are not correlated, as many small animals are very cancer resistant. This can be very applicable in human medicine. Now, I will summarize the contents of the primary literature. This is published in the National Library of Medicine. Many scientists, doctors, and veterinarians collaborated in this work. It was titled Potential Mechanisms for Cancer Resistance in Elephants and Comparative Cellular Response to DNA Damage in Humans. The study compared DNA reparation and apoptosis in human and elephant peripheral blood lymphocytes. The cancer mortality of elephants was found to be 4.81%, whilst human cancer mortality lies between 11% to 25%. Upon investigating the genomes of these 2 species, humans have 1 copy of the TP53 gene. They have 2 alleles of this gene. Elephants, however, have 20 copies of this gene. This means that they have 40 alleles. They also have 19 retrogenes of the TP53 gene, so they have 38 alleles of this gene. The TP53 gene codes for the p53 protein, which is the tumor suppressor protein. When p53 is inactivated, apoptosis is suppressed. There is a rapid increase in cells and the genome is rendered unstable. In response to DNA damage, the expression of the TP53 gene was increased in both humans and elephants, but it was much more prevalent in elephant cells. This expression was proportional to the copies of the gene. Thus, the cancer rate in elephant cells was much lower. Another protein found that was related to the prevention of cancer was P21. This protein stops cell cycles. Whilst the news article conveyed the general findings of the study, there were still some discrepancies between the 2 texts. The most obvious inconsistency is that the news article describes p53 as the tumor suppressor gene. In reality, it is the tumor suppressor protein. TP53 is the gene that codes for this protein. This was not the only issue with the news article. Additionally, it stated that humans have 2 copies of the tumor suppressor gene, but as mentioned in the primary literature, humans only have 1 copy of the gene. However, they do have 2 alleles, and that may have caused the confusion. The news article also stated that elephants have dozens of this gene, but they only have 20 copies of this gene and 19 retrogenes. An important factor that the article left out is the expression of the P21 protein in response to DNA damage. Even though it was only briefly mentioned in the primary source, it is still a factor that prevents the development of cancer. There were still accuracies within the news article that are worth mentioning. The most obvious being that the gene is responsible for this resistance. The mortality rates that were reported were accurate. The article stated that less than 5% of elephants die of cancer, and that is true to the primary source, as it was reported that 4.81% of elephants meet their ends due to cancer. The article also stated that up to 25% of humans die from cancer. This was the upper end of the mortality rate as sourced from the primary report. The lower end that was left unmentioned was 11%. Even though the news article conveyed the main point of the study, the incorrectly reported details have the potential to spread misinformation. It has been proven that news outlets are not credible sources of information. Knowledge is best obtained from primary sources and professionals within that area. This way, it is ensured that the information is accurate.